Welcome to Biochemistry Unfolded, From Atoms to Algorithms, an online podcast on the topic of biochemistry and biophysics. Hey, good afternoon, and welcome to another episode of Biochemistry Unfolded, From Atoms to Algorithms. Today, I'm exploring the interesting origins of how metals have come to be utilized and incorporated into living organisms. When we think of life, we think of 6 commonly associated elements in the periodic table: hydrogen, carbon, nitrogen, oxygen, phosphorus, and sulfur. I teach my students in biochemistry courses that the chemical symbols for these elements spells out schnapps, just as an easy way to remember these important elements. And it's quite easy to instantly make the assumption that life equals nonmetals and everything else equals metals, right? But in fact, that is quite far from the truth. So living organisms actually need metals for essential biological processes. We need iron to bind with oxygen and hemoglobin, and that enables respiration, and that's how we breathe. Copper is also an essential cofactor in, for example, the cytochrome C oxidase reaction in the electron transport chain, and that's essential for ATP synthesis, among many other things. Zinc is essential for our immune system to function. And of course, magnesium is an essential mineral required for over 300 biochemical reactions in the body. And that includes muscle and nerve function, blood pressure regulation, as well as energy production. So magnesium is also at the heart of chlorophyll molecules in photosynthesis. And that's essential for converting energy, light energy into chemical energy. Out of 118 elements on the periodic table, we have about 94 metals, so it's easily the vast majority. And many of these are certainly found in living organisms. Today, I'm actually going to focus on the evolution of 2 metals in early living organisms, and that's molybdenum and tungsten. These are both in group 6 of the periodic table. Molybdenum, Mo, is atomic number 42, and this is an essential trace metal in modern biology, and it resides at the catalytic core of enzymes such as molybdenum, and these drive critical biogeochemical cycles, including the carbon, nitrogen, and sulfur cycles. Molybdenum was discovered in 1778 by Carl Scheele, and the features closely resembled that of lead. The name molybdenum actually traces to the Greek word molybdos, meaning lead-like, and the essential role of molybdenum in human life was established in 1953 by DeRenzo, Richert, and coworkers, and they identified molybdenum as a cofactor of xanthine oxidase, which is also involved in the uric acid biosynthetic pathway. The ubiquitous pterin-based molybdenum cofactor MoCo is also part of the active centers of all molybdenum enzymes in living organisms, and without these, molybdenum would remain catalytically inactive. Now, the enzyme nitrogenase is used in bacteria such as rhizobium, and it's found in the roots of legumes. This has a specialized iron molybdenum cofactor, and it's involved in nitrogen fixation that converts atmospheric nitrogen, N2, into usable ammonia, or NH3, for amino acid synthesis. And plants and algae also use molybdoenzyme nitrate reductase to reduce soil nitrates from NO3 to nitrites NO2, and that's also used for amino acid synthesis. So molybdenum is definitely an essential metal for biochemical reactions. But geochemical data indicates that in early Earth oxygen-free oceans, the anoxic oceans, prior to the great oxidation event of about 2.45 billion years ago, dissolved molybdenum was extremely scarce. A recent paper published this month in Nature Communications examined this in more detail. And in this work, Klos and colleagues examined tungsten alongside molybdenum. Now, tungsten is atomic number 74, and this is also in group 6. It shares similar chemical properties, and it's involved as a cofactor in biosynthetic pathways with molybdenum. Tungsten enzymes are also typically favored by prokaryotes operating in anaerobic or oxygen-free environments. So the study actually found a distinct ecological divergence in how these metals are used today. Overall, aerobic organisms tend to possess more molybdenum-dependent genes, whereas anaerobic organisms display a stronger reliance for the tungsten-dependent genes. So the researchers actually examined the core question of how early life managed to evolve complex molybdenum-dependent metabolic pathways, such as nitrogen fixation, when the metal itself was incredibly difficult to source in the primordial environment of ancient Earth. The research team actually conducted phylogenomic screening first by assembling a comprehensive data set of about 102 protein groups representing the core molecular machinery for molybdenum and tungsten transport, as well as intracellular storage and cofactor biosynthesis. This actually included genes from the 4 major molybdo-tungsten enzyme families, as well as nitrogenase. So it's essential to start out with a good description of what we're looking for. Essentially, we can look at figure 1 in the paper, and we think of this as sort of a blueprint or a shopping list of essentially everything we need. Now, in bioinformatics, phylogenetic screening is like searching through the instruction manuals or a DNA of thousands of different organisms to find very specific parts or genes. By seeing which organisms have the instructions for certain parts and which don't, scientists can then trace the evolutionary history of those parts to see when they first appeared in the tree of life. But figure 1 itself isn't an evolutionary tree. Instead, it's just a biochemical schematic. That's a map showing the entire assembly line that a cell uses to handle these trace metals. It illustrates the uptake, the transporter proteins such as MOD-ABC that act as the doors to pull molybdenum and tungsten from the environment into the cell, and the trafficking and assembly, that's the internal worker proteins that modify the metal and build the complex molecular housing for it, such as the molybdopterin cofactor, or MoCo. And lastly, of course, we have the final product. That's the different families of mature enzymes that finally incorporate the metal to perform vital chemical reactions. Now, you can't simply screen a genome for molybdenum use as a broad concept. You have to search the DNA for very specific and tangible proteins that make that use possible. So we begin by visually defining exactly what we're looking for. By mapping out every step of the metal's journey from the outside environment to the active site of the enzyme, figure 1 breaks a complex biological process down into specific identifiable protein targets. And so the researchers looked at 102 of these. They took this exact list of proteins that are defined in the figure and screened the genomes of thousands of species to see when each specific door, worker, and enzyme first evolved. Essentially, this figure is the physical model of the machinery and the phylogenetic screening was the method used to figure out when each part of that machine was invented. So this concept can be kind of tricky at first because it requires linking a physical biochemical pathway directly to the computational genetics. Now, the next phase of the research involves sequence alignment and annotation. Now, they use traditional bioinformatic tools here, such as HMER, or HMMER, and the KOfam profiles. They screened genomes across the entire tree of life to identify candidate protein sequences, and then these sequences were actually aligned with MAFFT. These are fairly standard biochemical bioinformatics tools. HMER is a powerful search engine that searches through unknown genetic sequences to see if they belong to protein families. And specifically, it uses statistical probabilities to find matches, even if a gene has mutated or changed just so slightly over millions of years. And of course, the KOfam, is the massive categorized encyclopedia of these known protein families that each entry defines a specific biological function, and that's what HMER was used to analyze. So once these similar protein sequences were found, MAFFT acted as a meticulous organizer, and that lined them up side by side. And this process is also called multiple sequence alignment. And so scientists could see exactly which specific building blocks of the protein stayed the same and which changed through evolution. So next, the researchers performed a phylogenetic reconstruction. And so they constructed phylogenetic trees using FastTree, and that employed the WAG evolutionary model to map the evolutionary relationships and the origins of these proteins. So if figure 1 is like the shopping list of specific proteins that researchers are looking for, the phylogenetic reconstruction is the process of building the massive family tree to see exactly where those proteins actually lined up. So what is phylogenetic reconstruction? When scientists want to know how different species or specific genes are related, they compare their DNA or protein sequences. Organisms with very similar sequences are closer related, like siblings, while those with very different sequences share a much more distant ancestors. So we kind of have like a tree-like structure, right? Kind of like the branches of a tree. And so phylogenetic reconstruction is the computational math that's used to draw the branches of this family tree, tracing everything back to a common ancestor. So figure 2 is actually the visual masterpiece of this phylogenetic reconstruction. And it takes a massive pre-built family tree of life representing thousands of species of bacteria, archaea, and eukaryotes, and it maps the data that they found right onto the branches. Think of it like a giant ancestry chart where you've used different colored highlighters to mark specific traits. Figure 2 actually overlays two main things onto that tree. First of all, who has the tools? It highlights which specific branches or organisms actually possess the molybdenum or tungsten genes that we're looking for in figure 1. Number 2, we want to ask the question, where do they live? And so the figure actually colors the branches based on the organism's ecological traits. Most importantly, where they are aerobic, living with oxygen, or anaerobic, living without oxygen. And so by mapping these genes and the habitats onto the family tree at the same time, the researchers could suddenly see very obvious evolutionary patterns. Figure 2 visually proves one of the paper's main conclusions, that the branches of the tree that breathe oxygen are packed with molybdenum genes, while the branches that survive without oxygen rely much more heavily on tungsten genes. So that's a very important observation to account for. So essentially, Figure 2 takes a massive amount of genetic code and it turns it into a map, allowing us to see exactly how early life adapted to its molecular toolkit to survive in different environments billions of years ago. So lastly, the scientists use a technique of molecular clock dating. And so this estimates the specific geological eras when the genes that are responsible for molybdenum and tungsten utilization first emerged and duplicated. So the core idea behind a molecular clock is that mutations of DNA accumulate at a relatively steady rate over time, much like the steady ticking of a clock. If a specific gene mutates roughly once every million years or so, we see 10 differences between the gene in species A and species B. And then we can infer that they split from a common ancestor about 10 million years ago. So, of course, the biological clock isn't perfect. Some genes mutate faster than others, and environmental pressures can change the speed of the ticks. To fix this, scientists have to use little calibrations. And so they're going to look at the physical fossil record or geological events, like the sudden appearance of oxygen on Earth, to anchor specific points on the tree to absolute calendar years. Then we can use complex mathematical algorithms to calculate the timeline for the rest of the tree. So Figure 3 is where the researchers apply this molecular clock to the original shopping list of proteins. And so, that's what, we take Figure 3 and then cross-reference it with Figure 1, right? So instead of just showing how the genes, who has the genes today, like in Figure 2, Figure 3 adds a timeline to that tree. If you look at the figure, you notice that the horizontal axis on the chart is explicitly marked with geological timescales, and that spans from the Archean Eon, billions of years ago, right up to modern Proterozoic and Phanerozoic eras. So Figure 3 uses that molecular clock to tell the story of molybdenum and tungsten transport, first of all, by dating the nodes. So every time a branch splits on the phylogenetic tree, it represents a common ancestor. That's a node. And by applying the molecular clock algorithm, the researchers can put date stamps on the individual nodes that are responsible for mod ABC, that's your molybdenum, and TUP ABC, that's your tungsten, right? And those are the two different transport systems. So then the big reveal in Figure 3, this demonstrates visually that the roots of these transport systems track all the way back about 3.4 to 3.7 billion years ago. And so we have different gene duplications. Our timeline in Figure 3 also shows when genes duplicated to handle different metals. It illustrates even when the early oceans had almost zero molybdenum, the molecular clock places the origin of highly specific molybdenum grabbing pumps deep in the ancient Archean Eon. So in short, Figure 3 takes the evolutionary relationships and stretches them across a multi-billion year calendar. And that proves that life's machinery for capturing these rare metals is fundamentally ancient. And so in conclusion, our study provides molecular dating evidence that the biological utilization of molybdenum and tungsten stems back to the Eo-Meso-Archean eras, approximately 3.7 to 3.1 billion years ago. And so these findings fundamentally change the prevailing assumption that the scarcity of trace metals in the early oceans restricted the emergence of complex biochemistry. So instead of simply relying on elements that were locally abundant, early microbes evolved rather elaborate, highly specific molecular infrastructures to capture and harness trace amounts of molybdenum and tungsten. And so this highlights the profound antiquity, adaptability, and resourcefulness of early life. And it demonstrates that life's molecular toolkit successfully transcended environmental limitations to facilitate critical energy harnessing reactions billions of years ago. However, the research does not really answer every question. And in fact, it may open up new ones, right? So first of all, how reliable is a molecular clock that ticks back 3.4 billion years? That's an enormous amount of time. Molecular clock dating relies on statistical models of how fast DNA mutates, and it's calibrated by known events in the fossil or the geological record. However, the further back in time you go, that wider margin of error becomes a bit bigger. And so readers should critically ask, are the evolutionary models used here robust enough to definitively place these gene families in the Archean Eon, or could slight changes in the estimated mutation rates shift these origins by hundreds or millions of years? Number two, did these genes actually evolve from a common ancestor, or were they stolen? Bacteria and archaea are notorious for horizontal gene transfer, or HGT, and that's the ability to copy and paste useful genes directly into their neighbors, regardless of species. And so we need to ask, does the widespread presence of molybdenum and tungsten genes across different ancient lineages prove that they originated from a single, deeply ancient ancestor, or did a later organism invent them and rapidly share them across the microbial world, right? So the authors use phylogenetic reconstruction or reconciliation to account for this, but HGT also complicates deep evolutionary trees. Number three, does global ocean scarcity actually matter to a localized microbe? The paper frames its core paradox around the fact that the early oceans, on average, were severely depleted of dissolved molybdenum. However, a critical reader might ask, what if the first molybdenum-dependent microbes evolved in a highly localized, metal-rich environment like hydrothermal vents? If life originated in a localized chemical hotspot, then that global average of molybdenum in the ocean might be entirely irrelevant to the survival of those very specific early organisms. And number four, how did life physically survive the great oxidation event, which was essentially a metal swap? Around 2.4 billion years ago, oxygen flooded the earth. Tungsten suddenly became locked in the rocks and insoluble, while molybdenum became highly soluble and abundant. The paper actually notes the ecological divergence this caused, anaerobes keeping tungsten, while aerobes favored molybdenum, and the molecular mechanics of how microbes physically survived this massive transition in real time is left largely unanswered. So how did the internal molecular machinery seamlessly swap the preference from one metal to another without the cells dying in the process? So these are all important questions to think about. So thank you for watching this episode of Biochemistry Unfolded, from atoms to algorithms. Please like and subscribe to my channel to stay updated on new content. Hit the little bell icon, and that way you'll constantly get a notification whenever new content is posted. Do you have any questions, any other questions related to this paper or to bioinformatics analysis in general? Please leave them in the comments below, and I do read those comments. And if you have anything interesting to share, let me know about it. And until then, I will see you in the next video. Thank you for watching. You have been listening to Biochemistry Unfolded, from atoms to algorithms. Thank you for watching. We will see you again in the next video.